Okay, as we get started, here's our equipment. We've got everything set out. You'll see that we've got a centrifuge, we've got our glassware, we've got two cotton tipped applicators or swabs, we've got our pipettes, a marker, some small centrifuge tubes. This one contains water, 1 milliliter of water. We'll use that with the centrifuge. This one contains 1 milliliter of lysis buffer that's already in there. We have our methylene blue. We're going to use that at the end. There's the label for it, methylene blue. It's a stain. We've got our salt or NaCl, sodium chloride solution. We've got our protease. It's written on there. It's kind of blurry. Protease is an enzyme solution that breaks down proteins. And that is our tube that we're going to actually use to collect our sample a little bit later on. And this is a little float for the water bath. This is the little tube that we'll put our final sample in when we're finished, and then we're ready to go. Alright, so the first thing we're going to do is we're going to put some cells in our lysis buffer. I've labeled it PL, and we're going to, it would just be your initials. We're going to use our swabs to get our cells from our cheek. It's very easy. All you have to do is rub the inside of your cheek with a cotton tipped swab. There are two in the package, and we'll use both of them to make sure we get lots of cells. So I'm just rubbing the inside of my cheek. And you don't have to rub real hard because those cells come off pretty easily. And I'm now going to stick the tip of the applicator into the lysis buffer, kind of swirl it around, and that'll get the cells off of the applicator and into the lysis buffer. And as mentioned at the beginning of the video, make sure you know what all of these chemicals do. Why are we using them? Okay, we're going to do the same thing again. Another applicator. And swirl that around in there, dislodge the cells, kind of squish it around so that the cells will be released into the solution. Okay, and we're done with those. And they need to go in the trash. Okay, and now we have our cells in lysis buffer. Okay, now we need to add some protease because as those cells are sitting in that solution, we need to break down the enzymes and proteins that might damage the DNA. And it also will allow the DNA to be released from the histones. So just a few drops. The exact amount doesn't matter, but a few drops. Okay, and that should work. Okay, and we're done with the protease. And now we need to give the enzymes a chance to work, and we're going to put it in a water bath. So our little float there will just keep it suspended in the water bath for 10 minutes. Okay, we're back, and we've had a chance to let that incubate. We're ready now to use the centrifuge. So we're going to pop that open. And we're going to set the tube in there. Now remember that a centrifuge needs to be balanced, and that's where our little tube of water comes in. So this is 1 milliliter of water. We're going to put it across from our sample, and they need to have about the same amount of liquid in them so that they're the same weight and mass. And you'll see that they're opposite each other, and the hinges are pointed out. And that's going to keep the centrifuge from getting off balance. Oh, and we almost forgot our lid. So we need to put the lid on there, keep anything from flying around because it spins really fast. Okay, we're going to make sure that our speed is at 14,000 RPMs. And we're going to set the timer for 5 minutes and let it go. Okay, the centrifuge has shut itself off and we're ready to take out our sample. So we just open it up, make sure it stops spinning. And let's see, that one is our water, so we don't need that anymore. And that one is the one that has my initials, so we're going to keep on going with this. If you can see there, it's hard to see because it's blurry, but there is a white kind of a blob on the bottom of the tube. That's the pellet, and that's where all the cell debris has collected. What we want to do is put the supernatant, the liquid, into our new test tube. So we're extracting the liquid but not dislodging the pellet at the bottom. And again, it doesn't have to be all of it, we just want to get most of it, but we don't want to get the stuff at the bottom. We don't want other organelles and other cell parts. Okay, so we're done with that. That's basically just the pellet and a little bit of liquid left. Our supernatant is now in our tube, and that contains the DNA. We're now going to add salt, or sodium chloride, which is an ionic compound. You should know that. And again, we just need a few drops. That makes the solution ionic. And the DNA doesn't want to mix with an ionic solution. That will start causing it to form a precipitate, and you'll see it start to make a white stringy material in the solution. And it takes a little while for that to work. Okay, we'll get that out of the way. Okay, next we're going to add some alcohol, and what we need to do is make sure the alcohol is really, really cold. So I just got it out of the freezer, and we're going to add that carefully to the top of our sample. And we don't need a whole lot, and it doesn't have to be an exact amount. I'm just going to pour about a milliliter into a beaker here. Again, the exact quantity doesn't matter. That's about a ml. And then we're very carefully going to add this alcohol to the top of our salt solution. We just want to kind of let it run down the side. We don't want it to mix. We want it to make a layer on top. So I'm putting it in slowly and gently. You can see it's just kind of going down the side of the tube. And if you could see it up close in person, you'd be able to see that there's actually 2 layers of clear liquid. They kind of refract the light differently, but it's very hard to see in a video, especially if it's blurry. The DNA will collect between the 2 layers. It doesn't want to mix with the nonpolar alcohol, but it also doesn't want to mix with the ionic salt solution, so the DNA is going to form in a layer in between. We're going to wait a few minutes, and after about 5 minutes we should be able to see some white material somewhere in the middle of our tube. You can see it there. It's kind of looks like a little white glob. And that is our precipitated DNA. Okay, we're going to go ahead and add some methylene blue to that. That'll help make it show up a little bit more and stain the DNA blue. So I'm going to get a little transfer pipette, and we're just going to add really 1 drop. Okay, so a little bit of methylene blue, and that methylene blue will bind to the DNA, and it'll also diffuse somewhat into the solution, but our goal is for it to have a chance to bind to the DNA. Okay, it's been about 3 minutes, and you can see it's spread around a little bit in there. What we want to do is try to extract the clumpy blue stuff. So we've got another clean another clean transfer pipette. We're going to suck out what looks like the bluest, darkest blue material and transfer that to our new tube, which will be our new solution of DNA. A nice blue color. And there it is. We have isolated the DNA from the rest of the cell. A very common procedure in a lot of biology these days. Okay, and we're going to make it into a little necklace. We can add a lanyard to it. Lanyard is just a little plastic string kind of thing that We can put in the hinge of the tube, and just for fun, if you wanted to make it into something like a necklace, it has a little bit of your own personal, unique DNA in it. And that's it. We're done.